Hey guys, this is Mr. Bennett. This is notes number 14, and this is video 14C, meaning it's our third video section for these notes. Once you're done with these, you're done with notes 14. Our topic for this is going to be osmosis, and as we said earlier, osmosis is the second type of passive transport. I also mentioned that osmosis is really just a different type of diffusion. So in fact, it's the diffusion of the solvent, and we said earlier that the solvent in cells is typically water. So what that means is that the water is moving from an area of high to low. Now, when does this occur, or why does this occur? It generally occurs when the solute is unable to move. In other words, solute, which would be things like salt, could be protein inside the cell, could be sugars inside the cell, whatever your solutes are, okay, they're unable to move. They're unable to get through that semi-permeable membrane. And so instead of the solutes moving, the water will move, but the goal is still the same. The goal in both cases is equilibrium. Standard diffusion just moves the solute, it would move the salt, the protein, the sugar, etc. to get to equilibrium. In this case, water has to move to get to equilibrium. So in order to understand osmosis and the way that water moves, we got to think about the high and the low. So just like diffusion, you're moving from high to low, but in this case, it's the high water concentration to the low water concentration. And these are a little difficult to visualize. So what I mean by high water concentration is it's the side or the area with the least amount of solutes compared to the water. So if we were to draw a cell, and we're going to put lots and 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 lots of proteins or solutes inside this. Okay, and then we had a few proteins or solutes on the outside, but not many. Okay, the side with the least amount of solutes is the outside, right? So outside the cell right now, we have a high concentration of water, meaning we have lots of water compared to the amount of solutes. There's a special name for this. We call this hypotonic. Okay, and the way I remember it is the O in hypo rhymes with low. So this is the side with the low solutes. Okay. Now the other side of the membrane, meaning inside the cell, is what we call hypertonic. This is the side that has the low water concentration. Okay, it's low in water. Well, what do I mean by that? I mean that we have lots and lots and lots and lots and lots of solutes in here and not as much water compared to the outside. So we would call the inside of the cell right now hypertonic. And the way I remember this is that when I'm hyper, I have lots of energy. So hypertonic means it has lots of solutes. So the side of the membrane that has lots of solutes we call hypertonic. The side that has very few solutes, or the low side, is hypo. So low for hypo, hyper means lots. Okay. Once we've labeled the two sides and we know that the inside is hyper and we know the outside is hypo, water will always move from hypo to hyper. So if I redraw my cell, we said that the inside was hyper, right? There we go. And we said that the outside was hypo. So what will happen is that water will move into the cell. This cell is going to fill with water. Okay, the inside is going to be diluted as water gets added in, and as we lose water from the environment is going to get concentrated, meaning its concentration will go up, and the concentration of water inside the cell will go down. Okay, or not concentration of water, but the concentration overall inside the cell will go down as water gets added and it gets diluted. Okay. Now osmosis is a little tricky, but the more you practice it, the more you practice labeling the hypo and the hyper side, remember it's hypo to hyper, you'll get it. Okay, so let's talk about what osmosis does to cells. We already talked about it in that picture, the idea that that cell was going to gain water through osmosis. So when it comes down to it, cells can either gain or lose water through osmosis. They generally gain water when they're in a hypotonic environment. Remember, it's always hypo, meaning water moves from hypo to hyper. So if the environment is hypo, that means water is going to leave the environment and go into the cell, so it gains water. If the environment is hyper, that means the cell would be hypo, So the cell itself inside would be hypo, and then water would leave the cell into your hyper environment. Okay, so let's look at some examples here. Kind of erase my pictures there, but that's so we can see these pictures without anything getting in the way. So on top we have animal cells. Okay, so this would be an animal cell in a typical body type environment. It's round, water's going in or out. Okay, we call this isotonic. Isotonic is really just a way of saying that we're at equilibrium. But when we're dealing with osmosis, we call it isotonic, meaning we have the same amount of solutes outside as we do inside. If I take this animal cell and I put it into a hypotonic environment, it will gain water. In fact, water will go in so much that if the hypotonic environment is too hypotonic, my cell will actually pop. Okay. Now the opposite would be to put it in a hypertonic environment. In this case, the cell itself is hypo, and we know that water moves from hypo to hyper. So in this case, the cell is going to lose water, and it's going to shrivel up like a raisin. Okay, that's an animal cell. Okay, animal cells typically want an isotonic environment. Plants are a little bit different. Okay, plants have a cell wall which prevents them from popping. So what plants want to do, they actually want a hypotonic environment, because hypotonic environments are going to drive water into the cell, right? Because the outside is hypo, the inside is hyper, water moves into the cell. That's going to bring water into the cell, which is needed for photosynthesis. It's also going to inflate this cell. When we inflate a cell and we make it nice and firm, nice and full, we call that turgid, meaning it's just like plump. And so by plumping up the cell with water, it's going to help the plant to stand upright. Okay? Now, if you don't water your plant, meaning you don't have a lot of water in that extra, like that fluid that's in the plant, then you get closer to isotonic, which means that instead of water being driven into the cell, now water is kind of going in and out at the same rate. At that point, the cells are going to become flaccid, meaning they become a little soft. This is when we start seeing a plant start to wilt, because it's not plump. Water isn't being forced into the cell. It's kind of like wishy-washy. When it's fully wilted, when the plant has literally like fallen over and starting to shrivel up, that's the point at which you've lost so much water around the cells in the plant that now the outside is actually hypertonic. So it's hyper out here. The inside was hypo, and so water has left the cell and it starts to shrivel up, and at this point the cell is going to pull away from its cell wall. You can actually see it like stretching away, and it's going to die. So plants love this. They want to be plump. They want water moving in, so they want hypotonic environments. Our cells, on the other hand, they want isotonic, because that way we're not losing water, we're not gaining water. It's at an equal rate. We stay the same. Alright, that is the end of our video, 14C. It's the end of Notes 14. Make sure you complete the WISC, meaning the online questions. I'll see you tomorrow.